Hello, my dear students. Welcome you all to Sri Jain Rank Zone classes. I am your mentor and your friend for the subject biology, and I will be taking the zoology part, that is animal science. My dear students, in the last lecture, we have started the introduction of the nervous system. Today, moving with the lead of this chapter, that is neural coordination and integration, we will talk about the central nervous system. Central nervous system or CNS in all the vertebrates including humans, CNS is dorsal. It is dorsally placed. Central nervous system is dorsal. It is hollow and it is non-ganglionated, while in invertebrates when present it is ventral, solid, double and ganglionated. So in all the vertebrates including humans, CNS is dorsal. It is hollow and it is non-ganglionated, while in invertebrates, when present, it is ventral, solid, double, and ganglionated. CNS is formed of two parts. First is brain, which is the upper and broader part lying in the head. Second is spinal cord, which is a lower, long, and narrow part running from beginning of the neck to the trunk region. So this part having brain or head is known as what? Cephalic part. And the spinal cord, it runs from neck to the trunk, that is abdominal region. CNS, central nervous system, is covered by three meninges and its wall has two types of matter. It is covered by three membranous coverings known as what? Meninges, and it consists of two types of matter. CNS vertebrates, it includes two types of matter. First is gray matter and the second one is white matter. Gray matter, it is formed of cell bodies. It is non-medulated nerve fibers. It consists of non-medulated without myelin sheath nerve fibers. It consists of neuroglial cells. It consists of dendrites of association neurons and motor neurons. So the gray matter, it basically consists of cell bodies, that is soma or cyton. Cell body is also known as perikaryon. It consists of non-myelinated or non-medulated nerve fibers. It consists of neuroglial cells. It consists of dendrites of association neurons and motor neurons. White matter, it is formed of medulated nerve fibers or myelinated nerve fibers. Myelinated nerve fibers or myelinated axon of motor and sensory neurons. It doesn't include the dendrites of association neurons and motor neurons. It includes axonal part, which appear white due to the presence of medullary sheath or myelin sheath. So gray matter and white matter, these are two types of matter which is present in the CNS or central nervous system of vertebrates. In brain, gray matter is outside and white matter is inside. While in case of spinal cord, because of crossing of fibers, the gray matter is inside and white matter is outside. Gray matter inside and white matter outside. Next slide. Meninges. The meninges are the connective tissue membranes which surround the brain and spinal cord of central nervous system. In fishes, there is only one meninx called meninx primitiva or pia mater. In amphibians, reptiles and birds, the brain is covered by two meninges or membranes. The inner pia arachnoid or outer dura mater. Inner is basically consisting of pia mater and arachnoid, and the outer is dura mater. In mammals, CNS is covered by three meninges or cranial meninges. Brain meninges are continuous with spinal meninges. So in fishes, only one, that is pia mater. Amphibians, reptiles, and birds. Two meninges. Pia mater and arachnoid are fused. Sorry. And outer is dura mater. And in case of mammals, it consists of pia mater, arachnoid, and dura mater. The three layers of cranial meninges in order from superficial to deeper. The outermost is dura mater, then middle one is arachnoid, and innermost is pia mater. Okay, so the outermost is dura mater, middle is arachnoid, and pia mater is innermost. Dura mater is non-vascular. It is tough, made up of fibrous connective tissue. Arachnoid matter is made up of reticular connective tissue with collagen and elastin fibers, while the innermost vascular pia mater, which is nutritive, made up of loose areolar connective tissue. Okay, so the dura mater is made up of fibrous connective tissue. It is non-vascular. It is tough. Arachnoid is made up of reticular connective tissue having collagen and elastin fibers. Innermost vascular pia mater, which is nutritive. It is made up of loose areolar connective tissue. Between dura and arachnoid matter presence of subdural space. No cerebrospinal fluid in mammals is present here. In mammals, especially the cerebrospinal fluid, it is not present in subarachnoid space, subdural space, which is between dura mater and arachnoid. Between arachnoid and pia mater is subarachnoid space, which consists of, or which includes cerebrospinal fluid in mammals. CSF also found in ventricles and central canal of spinal cord. So CSF is present in subarachnoid space in mammals. It is present in central canal of spinal cord, and it is also found in ventricles of brain. Between dura mater and periosteum, there is epidural space, an extension of dura mater between the two cerebral hemispheres called falx cerebri. So falx cerebri is basically extension of dura mater between two cerebral hemispheres known as falx cerebri. Tentorium, which is an extension of dura mater between cerebrum and cerebellum. So dura mater is having two types of extensions. The first extension is between two cerebral hemispheres, which is known as what, falx cerebri, and the extension between cerebrum and cerebellum is known as what, tentorium. Okay, so I hope this slide is clear to everyone. These are brain meninges. You can see the superior sagittal sinus. It is very important, superior sagittal sinus here. This is subdural space just beneath the dura mater. This is arachnoid, the white one, and this is subarachnoid space, and this is the pia mater. This is the dura mater. Okay. It is the skin of scalp. This is periosteum. These are the bones of skull. This is the covering of the bones of skull known as what? Periosteum or periosteal. Then these are meninges. So dura mater is basically divided into two parts. One is the periosteal covering and the second one is meningeal covering. This is the arachnoid meta and the third one is pia meta. Pia mater is vascular, non-vascular. Falx cerebri is the extension of pia mater between two cerebral hemispheres in longitudinal fissure only. Okay, sorry, extension of dura mater, not pia mater, extension of dura mater. Okay, cerebrospinal fluid or CSF. All the ventricles of brain, central canal of spinal cord, are continuous and lined by a columnar ciliated epithelium known as ependymal cells or ependyma. Very important. They contain lymph-like extracellular fluid called cerebrospinal fluid. This fluid is secreted by choroid plexus by filtration of blood. All the ventricles of brain, central canal of spinal cord are continuous and lined by a columnar ciliated epithelium known as what? Ependyma or ependymal cells. They contain lymph-like extracellular fluid known as cerebrospinal fluid. This fluid is secreted by the choroid plexus by filtration of blood. The choroid plexus consists of loose connective tissue of pia mater. As you know, dura mater, arachnoid, and pia mater, they all made up of different types of tissues. Okay. The outermost part is dura mater, the middle is arachnoid, and the inner is pia mater in case of mammals. Okay. So you have seen dura mater is made up of fibrous connective tissue. Arachnoid is made up of the collagen fibers and elastin fibers and reticular connective tissue, and the pia mater is made up of loose areolar connective tissue. So the choroid plexus consists of loose connective tissue of pia mater covered internally by a simple cuboidal epithelium of secretory or glandular nature. The choroid plexus it consists of the loose areolar connective tissue of pia mater covered internally by a simple cuboidal epithelium of secretory in nature, that is glandular in nature. The CSF or cerebrospinal fluid slowly flows toward the fourth ventricle by secretional pressure or secretion pressure and passes into the spinal cord. The cerebrospinal fluid flows towards the fourth ventricle of brain by secretion pressure and passes into spinal cord. Okay. Some fluid escapes into subarachnoid spaces through three pores, a median aperture of Magendie, and a paired lateral aperture of Luschka in the roof of fourth ventricle in the medulla. Some of the cerebrospinal fluid escapes into subarachnoid spaces through three pores. One is a median aperture of Magendie, and there are two pores which are laterally placed or lateral apertures known as what? Foramen of Luschka in the roof of fourth ventricle in the medulla. From the subarachnoid spaces, the cerebrospinal fluid is transferred to the blood of venous sinuses. Nervous tissue is without lymphatic vessels. Nervous tissue is not having any lymphatic vessels. Okay? The fluid escapes into the subarachnoid spaces through three pores. One is the median aperture known as what? Foramen of Magendie, and the second one is, and the two lateral paired apertures known as what? Foramen of Luschka. In the roof of fourth ventricle in the medulla, from the subarachnoid spaces, the cerebrospinal fluid leaks out in the blood of venous sinuses. Nervous tissue is without lymphatic vessels. Okay. Functions of cerebrospinal fluid. The main function is the protection to brain from mechanical shocks or physical injury. So is it basically act as shock absorber. Shock absorber. Okay. It helps to protect the brain from mechanical shocks and physical injuries. That's why it acts as shock absorber. It helps in maintaining the optimum physiological fluid environment for neural functions, that is conduction of nerve impulses, transport of amino acids, sugars, and oxygen. It helps in the conduction of nerve impulses, transportation of amino acids, sugars, and oxygen. Okay. It helps in relief mechanism from increase in intracranial pressure that occurs with each arterial pulse of the blood to brain. It helps in relief mechanism for the increase in intracranial pressure that occurs with each arterial pulse of blood to brain. It helps in sink-like facility for metabolites of brain. So it helps in protection of brain. It helps to maintain optimum physiological fluid environment for neural functions like conduction of nerve impulse, transport of amino acids, oxygen, etc. It helps in relieving the excessive intracranial pressure. It helps in sinking like facility of metabolites of brain. The blood cerebrospinal fluid barrier or blood brain barrier for selective transport process between blood and CSF. So the blood CSF barrier helps in the selective transportation of the nutrients. It helps in nourishment to central nervous system. It helps to provide nourishment to central nervous system. Now, major site of CSF formation is choroid plexus and mid-ventricular wall and subarachnoid wall also contribute the formation of CSF. The major site is choroid plexus. Along with choroid plexus, mid-ventricular wall and subarachnoid wall also contribute in the formation of cerebrospinal fluid. The specific gravity of cerebrospinal fluid is 1.005. CSF is cellular free, it is acellular, slightly alkaline, and is isotonic to plasma. The rate of formation of CSF is 20 milliliters per hour, that is 480 ml per day. 20 ml per hour approximately, or half liters per day. Total amount present in and around CSF is 80 to 150 ml. It means there is at least 3 times renewal of CSF every day. CSF contains glucose, proteins, lactic acid, urea, Na+, K+, Ca2+, Mg2+, Cl- ions, bicarbonate ions, and some of the WBCs. Blood-brain barrier facilitate maintenance of stable internal environment. It acts as a physiological and pathological barrier. The CSF helps to provide buoyancy so that the weight of the brain cannot be felt. Okay. The actual weight of brain is 1.4 kgs, but because of CSF, it comes down, the relative weight comes down to 0.8 kgs. 2.8 kgs, right? Hydrocephalus. The enlargement of head, it is a pathological condition characterized by abnormal accumulation of cerebrospinal fluid, resulting headache, vomiting, pain, stiffness of the neck. Hydrocephalus is a pathological condition in which there is abnormal accumulation of cerebrospinal fluid, resulting in the headache, vomiting, pain, and stiffness of the neck. Increased cerebrospinal fluid may result in meningitis. Increased cerebrospinal fluid may result in meningitis. Meningitis may appear due to the infection and inflammation of meninges or injury to meninges. Meningitis may appear due to infection or inflammation of the meninges or injury to the meninges. Infection may be viral, bacterial, or both. The most common cause of meningitis is the infection of Streptococcus pneumoniae, Neisseria meningitidis, and Haemophilus influenza. So these bacterial infections are most common for meningitis, Streptococcus pneumoniae, Neisseria meningitidis, and Haemophilus influenza. Lumbar puncture is done for drainage of excess of cerebrospinal fluid during meningitis. Sorry, puncture is done for the drainage of excess of cerebrospinal fluid during meningitis. Cerebrospinal fluid is formed by choroid plexus, anterior choroid plexus, and posterior choroid plexus. Choroid plexus. There are three choroid plexus in humans. The lateral choroid plexus, it is the roof of first and second ventricle. The lateral choroid plexus is the roof of first and second ventricle. Anterior choroid plexus, it is the roof of third ventricle or diaceal. The lateral choroid plexus, it is the roof of first and second ventricle. The anterior choroid plexus or ACP, it is the roof of third ventricle, diaceal. And posterior choroid plexus or pleochoroidea, pleochoroidea, it is the roof of fourth ventricle. Oxygen and glucose requirements. Brain controls the functions of our body organs and also provides the qualities of mind like learning, reasoning, memory. For these activities, brain needs a large and constant energy supply. At any given time, the activities of brain account for 20% of the body's consumption of oxygen and 15% of its consumption of blood glucose. Oh my goodness. I mean, only brain needs 20% of the amount of oxygen, and 15% of the glucose consumption is done by brain because the brain needs to constantly maintain all these activities, integral activities, qualitative activities of mind like learning, reasoning, and memory. Brain deprived of oxygen for just 5 minutes is permanently damaged. Mental confusion results if brain is deprived of glucose. This is the secretion of CSF. This is the subarachnoid space. The subarachnoid space receives the CSF through foramen of Magendie and foramen of Prushka. These are arachnoid granulations. This is dura mater. This is aqueduct of Sylvius. This is fourth ventricle where it is formed actually with the help of anterior choroid plexus, posterior choroid plexus, and lateral choroid plexus. Cisterna magnae. This is the central canal. It helps to maintain the continuity. This is third ventricle, interventricular foramen, septum pellucidum, and choroid plexus of lateral ventricles. So choroid plexus of lateral ventricles, these are, you can see the ciliated or, the cells having these projections. Structure of human brain or encephalon. Now we are moving to structure of human brain, encephalon. So I believe the previous slides were they are comfortable and they are being understood by you. Structure of human brain. Brain is also known as encephalon. It is a soft, whitish, large-sized and slightly flattened structure present inside cranial cavity of cranium of the skull. In men, it is about 1200 to 1400 grams in weight and has about 10,000 million neurons. Brain is made up of three parts. It is a soft, whitish, large-sided and slightly flattened structure present inside cranial cavity of cranium of the skull. In man, it is about 1200 to 1400 gram in weight. It has about 10,000 million neurons. Brain is made up of three parts. Forebrain or prosencephalon. It forms anterior two-thirds of brain and is formed of three parts. Forebrain or prosencephalon. It forms anterior two-thirds of the brain and it is formed of three parts. Olfactory lobes. They are in a form in pair. They are one pair, small sized, club shaped, solid, completely covered by cerebral hemisphere at the dorsal side. Okay. So olfactory lobes, they are paired, they are small size, they are club shaped, they are solid and completely covered by cerebral hemisphere on the dorsal side. Each is differentiated, mean each part, as they are paired, so each is differentiated into two parts. One is olfactory bulb, which is anterior and swollen part, and the other one is olfactory tract, which is posterior and narrower part, which ends in the olfactory area of temporal lobe of cerebral hemisphere. So the olfactory lobes have two parts, the anterior placed olfactory bulb and the posterior placed olfactory tract. Olfactory tract merges into the olfactory area of temporal lobe of cerebral hemispheres. Okay. The anterior part is swollen, okay, and the posterior part is narrow. Functions of olfactory lobes. They control the smell. It is normal in frog, rabbit, and man. It is well developed in dog, so power of smell is more in dog. These are also well developed in dogfish. Name dogfish is on the basis of well developed olfactory lobes. You can see this part is cerebrum. This is the pituitary gland. This is the posterior part. This is the anterior part of the body, especially head, cephalic index. This is the spinal cord which is running down. This is cerebellum. This is medulla oblongata, which continues to form, the hindbrain part of the brainstem. Pons, varolai, midbrain, pineal gland, hypothalamus, epithalamus, which are the part of diencephalon, thalamus, epithalamus, hypothalamus, pineal gland. Okay. Cerebrum. Cerebrum is divided into five lobes: frontal lobe, parietal lobe, occipital lobe, temporal lobe, and insula. So frontal lobe, parietal lobe, occipital lobe, temporal lobe, and insula. A lobe called insula is hidden as it lies deep in the sylvian fissure. It lies deep in the sylvian fissure. The cerebral hemispheres are separated from olfactory lobes by a rhinal fissure. So these cerebral hemispheres are separated from olfactory lobes by a rhinal fissure. The median fissure divides the cerebrum into right and left cerebral hemisphere. So which fissure divides the cerebrum into two cerebral hemispheres, right and left? Median fissure. Sylvian fissure is basically the insula, which is a deep hidden lobe, okay, in the sylvian fissure. The cerebral hemisphere is separated from the olfactory lobes by rhinal fissure. Few sulci which are well developed and form three deep and wide fissures which divide each cerebral hemisphere into four lobes: anterior frontal lobe, middle parietal lobe, posterior occipital lobe, and lateral temporal lobe. Frontal lobe is at the anterior, middle is the parietal lobe, posterior is the occipital lobe, and lateral is the temporal lobe. Fissure sulcus lying between frontal and parietal lobes. Frontal and parietal lobes, they are separated by, you know, the fissures lying between the frontal and parietal lobes is the central fissure or the sulcus. Okay, so central fissure or the sulcus is basically between the frontal and parietal lobes. That lying between the parietal and occipital lobes is parieto-occipital fissure. Between parietal and occipital is parieto-occipital fissure, and that demarcating frontal and parietal lobes from the temporal lobe is lateral or sylvian fissure. So sylvian fissure or the lateral fissure, which basically demarcates the frontal and parietal lobes from the temporal lobe. So the frontal lobe and the parietal lobes, they are basically separated by the central fissure or the sulcus. Okay, parietal and occipital lobes from parieto-occipital fissure, and they are demarcated from frontal and parietal lobes from sylvian or lateral fissure, which separates temporal lobe from frontal and parietal lobes. Each cerebral hemisphere is with a fluid-filled cavity called lateral ventricle or paracele. Each cerebral hemisphere is with a fluid-filled cavity, which is known as what? Lateral ventricle or paracele. Okay. So, cerebrum is divided into five lobes: frontal lobe, occipital lobe, temporal lobe, insula, and parietal. Parietal and occipital are separated by parieto-occipital fissure. Frontal and parietal lobe, they are separated by central fissure or sulcus. Frontal, parietal, and occipital, they are separated by temporal lobe through the sylvian fissure or the lateral fissure. Okay. Now, two cerebral hemispheres, they are also separated by they are also separated by median fissure. Okay. Two cerebral hemispheres are interconnected by a thick band of transverse nerve fibers of white matter called corpus callosum. The corpus callosum basically separates two cerebral hemispheres, which is the band of transverse nerve fibers made up of white matter. The peripheral portion of each cerebral hemisphere is formed of gray matter, means outside, and is called cerebral cortex, while the deeper part is formed of white matter, that is known as what? Cerebral medulla. Cerebral cortex is the highest center for many sensations and activities and is with a number of sensory areas. Cerebral cortex is 2 to 4 millimeters in thickness. Two cerebral hemispheres are interconnected by thick band. It is already, I think this part is repeated. No issues. Histology of cerebrum. The whole brain possesses gray matter outside and white matter inside around ventricles. Gray matter in cerebrum, gray matter is very much developed. It is on an average 2 to 4 millimeter thick, but at poles its thickness is 1.3 millimeters. It is thickest at the precentral gyrus, 4.5 millimeter thick. Gray matter of cerebrum is called cortex or pallium. Phylogenetically or evolutionary, cortex is divided into three parts. So gray matter of cerebrum is highly developed and it is thickest at the precentral gyrus, 4.5 millimeter thick. The average thickness is 2 to 4 millimeters, but it is thinnest at poles, 1.3 millimeter, and thickest at precentral gyrus. The gray matter of cerebrum is known as what? Pallium or cortex. Phylogenetically or evolutionary, cortex is divided into three parts: allocortex or paleocortex. It is the cortex of olfactory area of frontal lobe and olfactory bulbs. So allocortex or paleocortex, it is the cortex of olfactory area of frontal lobe and olfactory bulbs. In lower vertebrates, cartilaginous fishes, olfactory lobes occupy most of the part of cerebrum. So in these animals, sense of olfaction is very much developed. Sense of olfaction is oldest sense. Allocortex or paleocortex is highly developed in cartilaginous fishes. Okay, it is the cortex of olfactory area of frontal lobe and olfactory bulbs. Mesocortex, it is relatively not much older in development. Neocortex or neopallium or isocortex or neocephalon. It is the most recent cortex and is developed maximally only in humans. It is in prefrontal cortex or prefrontal region, organ of mind, precentral and precentral gyrus. The neocortex is having 6 layers of neurons, while remaining cortex only have 5 layers of neurons. So allocortex or paleocortex and mesocortex, they have 5 layers of neurons, and neocortex is having 6 layers of neurons. Okay. Neocortex or neopallium or isocortex. This is the most recent cortex and is developed maximally in humans. It is a prefrontal cortex or a present in prefrontal gyri region. Okay. The cerebral cortex is having an area of about 2200 centimeters square, while the cranial cavity is only 1450 centimeter cube. So to accommodate cerebrum, these appear foldings in the cortex. The ridges are called gyrus or gyri or convolution, while the depressions are called sulci or sulcus. The ridges are called gyrus or gyri, okay, like this. Okay, so these depressions are sulcus and these elevations are gyrus or gyri. Okay, this is the cortex region and this is the medullary region. Okay, so to hold this large amount of area of cerebral cortex, they undergoes infoldings and elevations. The infoldings or the depressions are known as what sulci, and the elevations or the convulsions or the ridges are known as what gyrus. So allocortex, mesocortex, and neocortex. White matter. It is the inner part of the brain. White matter is aggregation of myelinated and unmyelinated axons of marine neurons. Its fibers are divided into three categories. Commissural fibers. These neurons connect gyri of two hemispheres, such as corpus callosum, habenular commissure, anterior commissure, and posterior commissure. Commissural fibers. The neurons connect gyri of two cerebral hemispheres, such as corpus callosum. So they basically connects the two gyri of two cerebral hemispheres, such as corpus callosum. Habenular commissures. Other than corpus callosum, we have habenular commissure, anterior commissure, and posterior commissure. Then association fibers, they connect gyri of same hemisphere. So gyri of same hemisphere are interconnected by the association fibers. Gyri of two different cerebral hemispheres are connected by commissural fibers. Projection neurons. They are in fact ascending and descending nerve tract. They connect one part of the brain to the other part of the brain or to the spinal cord. In spinal cord, they were called as column. Projection neurons, they are in fact ascending or descending nerve tracts. They connect one part of the brain to the other part or a part of brain to spinal cord. In spinal cord, they are known as what? Columns. Details of gyri, sulci, and fissures. You can see precentral gyrus, central gyrus. This is frontal lobe. This is parietal lobe. This is parieto-occipital sulcus on medial surface of hemisphere. This is occipital lobe. This is lateral sulcus. This part is temporal lobe. This is transverse cerebral fissure. This is cerebellum, pons, medulla, spinal cord. These are gyri and these are sulci. Okay. This is white matter. This is gray matter. Cortex of gray matter. These are the fissures or deep sulcus. Fissures. You can see. Anterior view, longitudinal fissure. Cerebral veins and arteries covered by arachnoid. Left cerebral hemisphere. Left cerebral hemisphere. Right cerebral hemisphere. Occipital lobe. Parietal lobe. Frontal lobe. Associated structures of cerebrum. Associated structures of cerebrum, they have following specific structures. Subcortex, the nuclei on white matter It is a cluster of gray neurons in depth of white matter. They are formed in whole brain and are named differently. Subcortex, it is basically present beneath the white matter layer of white matter. The nuclei of white matter, it is a cluster of gray neurons in the depth of white matter. They are formed in whole brain and are named differently. Basal ganglia or central nucleus, there are several groups of nuclei in each cerebral hemispheres. The cerebral hemisphere also consists of subcortex, which is the nuclei on white matter. Nuclei on white matter means white matters they have inside the parts of gray matter cluster of gray neurons. Okay, basal ganglia or central nucleus, there are several groups of nuclei in cerebral hemisphere. Corpus striatum is the largest nucleus, consists of caudate nucleus and lenticular nucleus. Striatum is the largest nucleus, consists of caudate nucleus and lenticular nucleus. The lenticular nucleus is subdivided into putamen, which is outer shell, globus, pallidus, which is ball-like. Other structure functionally linked to sometimes considered part of basal ganglia are. So corpus striatum is the largest nucleus, which is further divided into which further consists of caudate and lenticular nucleus. Lenticular nucleus is further subdivided into putamen and globus pallidus. Putamen is outer shell and globus pallidus is inner shell, which is ball-like. Okay. Some other functions linked and sometimes considered part of basal ganglia of some areas are colostrum. Colostrum, it is the name given to gray matter present between insula and putamen. Between insula and putamen, colostrum. Epistriatum or amygdaloid body, it is the structure present at the end of caudate nucleus. So end of caudate nucleus, which is the part of corpus striatum, it basically consists of epistriatum or amygdala or amygdaloid body. Colostrum, it is the name given to gray matter present between insula and putamen. Red nucleus and substantia nigra, they are present in midbrain. Red nucleus and substantia nigra of midbrain. Subthalamic nuclei of diencephalon. So subthalamic nuclei of diencephalon, red nucleus and substantia nigra, epistriatum and amygdaloid body, okay, claustrum, they all are considered as the part of basal ganglia. Basal ganglia consist of corpus striatum, which is further consist of lenticular nucleus and caudate nucleus. Caudate nucleus, at the end of caudate nucleus, there is the presence of epistratum or amygdaloid body. Claustrum, it is a part which connects the insula and putamen, okay. The lenticular nucleus further divided into putamen and globus pallidus, which is ball-like, and putamen is on the outer side. It also consists of subthalamic nuclei of diencephalon. This is the basal ganglia or central nucleus. Okay, this is the basal ganglia. Caudate nucleus, globus pallidus, putamen, nucleus accumbens, amygdaloid complex, substantia nigra, red nucleus, and tegmentum. Okay, these are the parts of basal ganglia. Functions of basal ganglia. What are the different functions of basal ganglia? Caudate and putamen, caudate nucleus and putamen. Caudate nucleus, they are the part of corpus striatum, and putamen is the part of lenticular nucleus. Control large autonomic movements of skeletal muscle like swinging of arm while walking. So large voluntary movements, automatic movements of skeletal muscles like swinging of arm while walking is controlled by caudate and putamen. Globus pallidus control muscle tone for specific body movements. Globus pallidus consists of control the movement of specific body movements and also help to control muscle tone. Corpus callosum. It is the band of white neurons present between both cerebral hemisphere and connect them on medial surface. It is present only in mammals. It has anterior part genu, middle part truncus, and the last part splenium. So basically corpus callosum is also having three parts. The anterior part is genu, the middle part is truncus, and the posterior, the inferior part is, last part is splenium. Below corpus callosum, there are two fused bands of white neurons called fornings. Below corpus callosum, there are two fused bands of white neurons which are known as what fornings. Their anterior part is called column and posterior part is called crura. So column and crura, they are the parts of fornix. Fornix is a band of white neurons. Okay, these bands of white neurons are fused and they are present just below corpus callosum. Between column and genu, a membrane called septum lucidium or septum pellucidum is there. So septum lucidium or septum pellucidum, it is a membrane between genu and column. Column is the anterior part, okay, of corpus callosum, and genu is basically the part of corpus callosum which is anteriorly present. Okay, genu is the part of corpus callosum and column is the part of fornix. And between them is the presence of septum lucidium or septum pellucidum. Septum lucidium encloses a space called V5 or pseudocele because it is not possessing CSF. That is why it is known as what? Pseudocele. So, caudate nucleus and putamen, they control large automatic movements of skeletal muscle like swinging of arm while walking. You can see and you can feel it as well. Globus pallidus control muscle tone for specific body movements. Corpus callosum, it is a bind of white neurons present between cerebral hemisphere and connect them on the medial surface. It is present only in mammals. It is anterior part genu, middle part truncus, and the last part splenium. Below corpus callosum, there are fused bands of white neurons known as what? Fornix. The anterior part of the fornix is column and the posterior part is crura. Below column and genu, genu is the part of corpus callosum and column is the part of fornix. It is separated by septum pellucidum, yeah, septum lucidium. It encloses a space V5 or pseudocele. Pseudocele is not having CSF, that is why it is not what? False cavity. Limbic system. Limbic system is present, sorry, it is present on the inner border of cerebrum or the floor of diencephalon. So the floor of diencephalon or the inner border of cerebrum, it consists of limbic system. It is also called emotional brain or animal brain. Limbic system controlling emotion, animal behavior like chewing, licking, sniffing, docility, tameness, affection, rage, pain, pleasure, anger, sexual feelings, fear, sorrow, grooming. It has following structure. Limbic system present on the inner border of cerebrum and floor of diencephalon. It is basically known as emotional brain as it controls the emotions of the body like chewing, licking, sniffing, docility, tameness, affection, rage, pain, anger, docility, pleasure, sexual feelings, fear, sorrow, grooming. It has following structures. Cingulate gyrus. It is a region of precentral gyrus. The region of precentral gyrus, which is the part of limbic system, is cingulate gyrus. Hippocampal gyrus. It is a region of temporal lobe near colloso-marginal sulcus. These two structures are combinedly called limbic lobe. So limbic lobe basically consists of cingulate gyrus and hippocampal gyrus. Okay, cingulate gyrus and hippocampal gyrus. Campal. Amygdala or amygdaloid body, it is the end of caudate nucleus as we have discussed in the previous slide. Olfactory bulb, it is the posterior part of the olfactory lobe. These are the inferior or inferior on the anterior surface of the brain. Olfactory nerve ends in these bulb. Mammillary body, they are found in hypothalamus. Olfactory bulb and mammillary body, both are the center for olfaction. Dentate gyrus, it is in between hippocampus and parahippocampus. Anterior nucleus of thalamus is located on the floor of lateral ventricle. Anterior nucleus of the thalamus, it is located on the floor of lateral ventricle. The limbic system contains the following structures. First is cingulate gyrus and hippocampal gyrus. Together they form limbic lobe. Cingulate gyrus is the region of precentral gyrus. Hippocampal gyrus, it is a region of temporal lobe near closo marginal sulcus. Amygdaloid body is the end of caudate nucleus. Olfactory bulb, the nerve ends, olfactory nerve ends in these bulb. They are on the inferior anterior surface of the brain. Mammillary body, they are found in hypothalamus. Olfactory lobe and mammillary body, they are the centers for olfaction. Dentate gyrus, it is in between hippocampus and parahippocampus. Anterior nucleus of the thalamus, it is located in the floor of lateral ventricle. Okay, so We will be starting diencephalon as the floor of the brain in the next turn. Till then, bye bye. Take care. Stay safe. I hope everything is understood. Still, my doors are always open. If you want to ask anything, you can, put down your questions. Till then, bye bye. Take care. Stay safe. Stay healthy.